So GIA has a instrument development department. What capabilities do they contribute to this effort?
So, as my colleagues have previously discussed, diamond is a unique material with many challenges for characterization. Depending on the application, whether that be for a thermal solution, quantum sensing, or the biomedical field, custom instrumentation is required to provide accurate and detailed information about these specific characteristics. This is also the case for GIA's testing of diamonds as gemstone, where bespoke instruments have been developed by our in-house instrumentation group by a team of engineers and scientists. To that end, we have developed unique capabilities in mapping the distribution of diamond defects, since homogeneity is important for certain applications such as quantum sensing or optical window transparency. We have developed a high-resolution, three-dimensional hyperspectral imaging system, which non-destructively measures the distribution of photoluminescence defects, such as the NV center, within a diamond volume. And this is of particular importance to growers or application specialists which want to exploit the unique properties of diamond for a specific application where spatial distribution is important. So in the following image, this is an example dataset from the three-dimensional hyperspectral imaging system that we have developed at GIA. And what you are seeing here is 4 cross-sections showing the total stone, and then different cross-sections following either the XY, XZ, or YZ planes. And what this is showing us is the evolution of this CVD diamond as it grew. This is something that is very difficult to see with other techniques. And the colors basically here correspond to different defects within the diamond corresponding to different events. For instance, the red color is associated with growth interruptions, whereas the green color is the CVD diamond that was grown on top of the substrate, which here is shown in blue. At each pixel, we have a high-resolution spectra, which allow us to characterize the emitting defects in this volume. The following video shows how this diamond evolved in the reactor, the step flow growth that it is caused in a CVD reactor, incorporating different concentrations of defects as this diamond grew with varying conditions. And we are seeing this growing up from the substrate into the reactor plasma itself. Growers need to be able to understand how their growth regime morphs over time during growth, and how that changes the incorporation of defects, in particular nitrogen. When growing purer materials, it becomes much more obvious when reactor conditions are changing, and this technique can help in understanding which changes are important to monitor and avoid during synthesis. A significantly less measured property of photoluminescence is its lifetime, which you can think of as the time it takes for light to be emitted from the defect after excitation from a light source. This is useful as it can be used to evaluate the concentration of paired nitrogen atoms in the diamond, something typically only possible with absorption at relatively high concentrations. Any facility which has this capability typically can perform it on bulk samples with no localization, with the rarer ability to measure these as localized points. However, at GIA, we have developed the ability to measure this lifetime property in a three-dimensional volume to create localized 3D maps of this concentration. We are also expanding our capabilities to include absorption and Raman mapping, key for evaluating the optical transparency and strain properties across plates and wafers in high resolution. A particularly useful instrument that we have also developed in-house is a diamond inscription device, capable of marking a diamond which has applications in writing sample identification directly onto the surface of a diamond.
Explain to us what can be used on single crystal or nanocrystal or polycrystal diamond wafers.
We routinely evaluate single crystal diamonds produced for gem applications, whether they be natural or lab-grown. They often have varying shapes and sizes, where we can handle a variety of facet geometries and sizes from sub-millimeter to much larger samples. Some of our diagnostic techniques are also suited for investigating other forms of diamonds, such as nano, micro, and polycrystalline diamonds.
Okay, great. Because the growers out there now are growing both, and they're all trying to reach larger sizes. So this this is going to be very important to them. So just as a follow-up question then, what kind of samples are best to start with for meaningful baseline evaluation? What should people be sending you?
So representative CVD or HVHT blocks or plates are typically the easiest to measure. When using bulk analysis methods, the sample may need to meet certain specifications, such as substrate removal from CVD grown diamond or single sectors from high pressure, high temperature diamond. We will work directly with the clients to clarify these specifications to help ensure a smooth workflow.